COVID, reactivated herpes viruses, HERVs, human endogenous retroviruses, and Behçet's disease, Part 1. These are notes for January 28, 2025. This is the first part of a multi-part discussion on this topic. The goal of this particular YouTube series is to examine how COVID infections can reactivate a set of dormant pathogenic agents in tissues. And in this YouTube series, we will discuss how this can take place in an autoimmune condition known as Behçet's disease. We will also examine the utility of vitamin D receptor agonists as a means of suppressing one of the key upstream drivers of tissue pathogenesis, and that is a herpes virus, HSV1, herpes simplex virus 1. To set up the background for this discussion, I'd like to first emphasize a key paper which was published early in the COVID pandemic. Human herpes virus 6, human herpes virus 7, and Epstein-Barr virus reactivation in pityriasis rosea during COVID-19. Pityriasis rosea is a type of rash. In this case, it was a young man who developed a rash-like set of lesions in the back of his throat. And when these lesions were examined, they found reactivated herpes virus 6, 7, and Epstein-Barr virus reactivation in these lesions. Now, if you look on PubMed for a different human herpes virus, it's human herpes virus 1, which is known as herpes simplex virus 1, HSV1. If you look for HSV1 in Alzheimer's, you'll find 325 results. This is because HSV1 is now recognized as a potential or key driver of Alzheimer's. Now, in the bottom part of this slide, I'd like to discuss certain types of lesions in the oral cavity, gingival, which refers to the gums, gingival recessions. And this is where the gums are receding from the teeth, and these recessions are caused by herpes simplex virus in a patient with COVID-19 infection. So this emphasizes that COVID, by reactivating a herpes simplex virus, initiates lesions that are characteristic of the secondary infection. And that's using an epistemology which views the COVID-2 infection as the primary agent and the HSV1 as a secondary event. In this YouTube, we will now discuss the ramifications of HSV1 reactivation, particularly in the reactivation of human endogenous retroviruses, HERVs. To discuss these connections, as well as potential therapeutic avenues, I would first like to draw on this recent review, Transactivation of Human Endogenous Retroviruses, HERVs, by Viruses. What does the term transactivation mean in this context? To start this discussion, it's useful to draw from AI Overview. AI Overview provides a concise but overly limited definition of viral transactivation. Viral transactivation is a process where viral proteins, such as HIV-1 TAT or adenovirus EA1, act as transcriptional activators to significantly increase the expression of viral or host genes, usually by recruiting host machinery to specific viral promoters. These proteins are essential for viral replication and often operate in the nucleus. In contrast, here is a definition of viral transactivation from Wikipedia. First, a discussion of natural transactivation. Transactivation can be triggered either by endogenous cellular or viral proteins, also called transactivators. These protein factors act in trans, i.e., intermolecularly. HIV and HTLV are just two of the many viruses that encode transactivators to enhance viral gene expression. These transactivators can also be linked to cancer if they start interacting with and increasing expression of cellular proto-oncogenes. HTLV, for instance, has been associated with causing leukemia primarily through this process. Its transactivator, TAX, can interact with P40, inducing overexpression of interleukin-2, a cytokine, interleukin receptors, GM, CSF, and the transcription factor CFOS. HTLV infects T cells, and via the increased expression of these stimulatory cytokines and transcription factors, leads to uncontrolled proliferation of T cells and hence lymphoma. Artificial transactivation. This is the type of transactivation that cell and developmental biologists are usually thinking about. Artificial transactivation of a gene is activated by inserting it into the genome at the appropriate area as transactivator gene adjoined to specific promoter regions of DNA. The transactivator gene expresses a transcription factor that binds to specific promoter region of DNA. By binding to the promoter region of a gene, the transcription factor causes that gene to be expressed. Let us return to the review published in 2024, Transactivation of Human Endogenous Retroviruses by Viruses. Now in this wheel diagram, in the center of it, you can see certain herpes viruses such as HSV1 and Epstein-Barr virus. On the right, you see SARS-CoV-2. Reading the figure caption, Summary of HERV activation by viruses. Human endogenous retroviruses that are activated by the indicated viruses and virus families are listed along with the types of samples where HERVs are detected. The post-viral diseases that are associated with the viruses are also highlighted. These disease states are highlighted by the outer rim of this wheel diagram. If you'll look to the right, you see a code for this information, associated post-viral diseases, neurological symptoms, multiple sclerosis, cancers, type 1 diabetes, hepatic disease. On the left, I have highlighted a specific herpes virus, HSV1, which we will discuss further in this YouTube series. HSV1 is capable of transactivating HERV-K protein as well as HERV-W protein. These retrovirus proteins are pathologic, and they are known drivers, contributors to the following disease states: multiple sclerosis, MS, amyotrophic lateral sclerosis, ALS, Alzheimer's disease, AD, Parkinson's disease, PD, rheumatoid arthritis, RA, and long COVID. Now, adding CoV-2 to this complex jigsaw, and that is a term that British people use to refer to as a jigsaw puzzle. Looking at the various pathologic agents, whether they are proteins or viruses or pathways that become unbalanced because of these agents. These are all part of the jigsaw that produces these disease states. And this creates a challenge for the epistemology of pathogenesis, especially histopathogenesis. For instance, the issue of a HERV protein. Is it a host factor? Now that might seem to be a strange way to refer to a fossil virus, which has been incorporated into the human genome. But that is the crux of the matter. These HERV viruses are present in all human beings, and they're pathologic when they become reactivated. So why do some individuals have many decades of life without expressing the pathogenesis from these viruses, these fossil viruses, HERV viruses? What creates susceptibility for tissue pathogenesis? These are some of the key issues that need to be addressed. Because CoV-2, by reactivating HSV1, reactivates HERV-K protein expression as well as HERV-W protein. And on the right, you can see that CoV-2 by itself, independent of HSV1, can do this. So this is a double hit system where CoV-2 can reactivate these HERV viruses, their proteins, which are pathologic. In addition, it can augment the pathogenesis by reactivating HSV1, which in tandem with CoV-2 expresses these HERV proteins. So what are the transactivators of HERVs in long COVID? Now I would like to connect to a separate YouTube series on this channel where we discussed the hypermethylation of the GPX4 promoter. Recall that GPX4 is the acronym, the abbreviation for glutathione peroxidase 4. This enzyme utilizes glutathione to reduce peroxidated lipids as well as reactive oxygen species in cells that are undergoing oxidative stress. For instance, a viral infection or a therapeutic treatment such as hyperbaric oxygen. So methylation of the GPX4 promoter is potentially very pathologic, and it is occurring in long COVID and in ischemic reperfusion injuries. In addition, the vitamin D receptor, its promoter, can also become hypermethylated in infectious diseases such as COVID, and it can also become methylated in autoimmune diseases and cancer. So losing antioxidant capacity sets up tissues for increased lipid peroxidation, which are drivers of the programmed cell death pathway known as ferroptosis. So ferroptosis is a key element of this jigsaw puzzle. It is a pathologic process by which many tissues and blood cells can be damaged or eliminated as a consequence of viral infections or perhaps transactivations of HERV viruses in these pathological states. And this is pertinent to specific disorders such as Alzheimer's disease. So HSV1 in Alzheimer's disease, several recent references. HSV1 is a potential driver of Alzheimer's disease, unveiling the involvement of herpes simplex virus 1 in Alzheimer's disease, possible mechanisms and therapeutic implications. Role of HSV1 in Alzheimer's disease pathogenesis, a challenge for novel preventive therapeutic strategies. And on this YouTube channel, we have discussed the role of HSV1 and COVID, and a URL link to that YouTube is highlighted in green at the bottom of this slide. So we will draw from this literature and examine the cellular, molecular, and tissue aspects of pathogenesis in these neurodegenerative disorders and how the upstream drivers, particularly the reactivation of herpes simplex virus 1 and the downstream transactivation of HERV-K protein or HERV-W protein, contribute to the tissue and cellular pathogenesis which typifies Alzheimer's disease. Now, before proceeding with the rest of this particular YouTube, I would like to introduce this paper from 2010. I believe it is very important. It's one of the few papers on PubMed that illustrates that herpes simplex virus 1 can infect other cells in a nerve that are not neurons, glial cells. And one type of glial cell, which is highly neglected in terms of being a reservoir for HSV1, are cells of the perineurium. So what is the perineurium? The perineurium is a connective tissue sheet surrounding a nerve fascicle, which I will define in a moment. So this tissue sheet is composed of cells known as perineurial cells, and HSV infects perineurial glial cells. So if you look in the upper right of this slide, you'll see an exploded diagram of the structure of a peripheral nerve, such as a cranial nerve. An example would be the vagus nerve or the trigeminal nerve. These are cranial nerves, but they are part of the peripheral nervous system. And it is well known that HSV1 infects the trigeminal nerve and infects the cells of the neurons of the trigeminal nerve, which are located in the trigeminal nucleus. That's the standard model. Unfortunately, when you have standard models, that becomes dogma. And dogma is when people say, I've never heard of HSV infecting glial cells. It must be incorrect because I haven't heard about it. And this is one of the problems of translating preclinical models, example, rodent models of HSV1 infection, into neurology and psychiatry and now immunology, because this paper from 2010 demonstrates that perineurial cells can be infected by HSV1, and the perineurium is a site from which HSV1 can reactivate. Let me read from both the beginning and end of this long abstract. Herpes simplex encephalitis, HSE, is a rare disease with high mortality and significant morbidity among survivors. We have previously shown that susceptibility to herpes simplex encephalitis was host strain dependent, as severe lethal herpes simplex encephalitis developed after injection of human herpes simplex type 1 virus, HSV1, into the whisker area of DA rats, that's a type of rodent strain, where PVG rats, a different strain of rats, remain completely asymptomatic. In the present study, we investigated the early immunokinetics in these strains of rodents to address the underlying molecular mechanisms for the observed difference. Now moving to the end. Our data demonstrate the novel observation that genetically encoded properties of the host nerve and perineurial cells, recruitment of phagocyting cells, these are white blood cells like macrophages, together with the low expression of TLR2 and TLR9, these are toll-like receptors, in the periphery define the susceptibility to HSV1 entry into the nervous system. Now, in the upper right, you see an exploded view of a peripheral nerve. And peripheral nerves include cranial nerves. There are 12 pairs of cranial nerves in vertebrates. One pair, there is a right and left vagus nerve. This is the 12th cranial nerve. If you look at a cross-section of a peripheral nerve, you'll see that it contains bundles, and these are called fascicles. The term fascicle is derived from the Latin term fasces, which is a term for a bundle. Here we're talking about a bundle of axons, which are myelinated by Schwann cells, and that is a term used in neurobiology to refer to a wrapping around of an axon by a glial cell, and this is called a glial sheath or myelination. The purpose of it is to increase electrical conduction and to prevent electrical crosstalk between adjacent axons. Now, the outer portion of this fascicle, this bundle of axons, is a connective tissue sheet, and that is the perineurium. Between axons, inside this fascicle, there is an endoneurium. So we have endoneurium adjacent to the axons. All of that is encompassed by this sheath, illustrated with a black line. And these fascicles are then wrapped in an epineurium, and that is the outer part of the nerve, thus the term epi, to the outside. This paper discusses infection of the perineurial cells. They're part of the perineurium, and the roles of toll-like receptor 2 and 9 on the susceptibility of these cells to HSV1 entry and infection. Reactivation from this site in one of these rodent strains, rat strains, was found to make the rat strain susceptible for the infection to spread into the central nervous system, leading to herpes simplex encephalitis. Reactivation of herpes simplex virus 1, either from the trigeminal ganglion or from perineurial cells in the trigeminal nerve, can lead to the production of infectious virions, which can move through axonal transport either into the central nervous system or out to peripheral organs. And it is well known that HSV1, once it reactivates, can move to the oropharyngeal cavity and produce cold sores, or also on the lips. And in other cases, HSV1 can move to the eye and infect the cornea. This is called HSV1 infectious keratitis. So at the bottom of the slide are two papers that pertain to this. One is a novel use of vitamin D receptor agonists to treat an HSV1 infectious keratitis. It was a severe case. The cornea perforated, which allowed the iris to protrude from the cornea. Very dangerous situation for the eye. The patient was hospitalized, given standard of care acyclovir to suppress the HSV1 infection. A graft of an amniotic, a fetal amniotic membrane was grafted to the eye. That was also standard of care. When the patient was discharged, the HSV1 reactivated again, but throughout the body. So IV acyclovir was also given, an outpatient. But to bolster the innate and adaptive immune systems to help suppress HSV1, the patient was treated not with vitamin D3, cholecalciferol, but hydroxylated versions of it. 25 vitamin D3, which is calcifediol, and 125 vitamin D2, paracalcitol, a synthetic analog of calcitriol. And utilizing calcidiol-pericalcitol as adjunctive therapies, the infection was suppressed. The patient ended up with a corneal scar, a cataract. However, after a year of being maintained on calcidiol and pericalcitol therapy, the patient was able to undergo a corneal graft, and that, of course, removes the cataract because a new cornea has been grafted to the eye. And remarkably, the patient has now 80% normal vision in that eye. So I bring this up because the paper on the right, labeled 3, shows oral ulcers in an autoimmune disorder known as Behçet's disease. And this is common in a group of ethnic groups in the Middle East which have a specific HLA haplotype. So in Behçet's disease, oral ulcers are common. They become activated, and then with treatment they often go into remission. But in this patient, the patient had a flare of these oral ulcers as a sequelae to COVID. Fortunately, they were able to treat this woman, and I will go into the details of this case study in a subsequent YouTube. She was treated with vitamin D3. Her calcifidiol level, or 25 vitamin D level in her blood, which what people call vitamin D level, was 9 nanograms per mil, very low. She was immunodeficient in this critical immune hormone. By bolstering her 25D levels, boosting her, quote, vitamin D levels in her serum, the oral ulcers went into remission. Now, we will talk about these oral ulcers in Behçet's disease because they are frequently infected with HSV1, making it plausible or even likely that HSV1 is one of the key drivers of this ulceration. Now, how does this pertain to COVID? In 2020, a group in Croatia published this key paper, a theoretical paper. SARS-CoV-2 dissemination through peripheral nerves explains multiple organ injury. We now know that COVID-2 can be transported within endosomes within the nervous system. And so when mucosal tissues like the lungs, or the tissues that line the lungs and the gut, become infected with COVID, the infection can be amplified there, and virions can move from the mucosal tissues into neurons that innervate these mucosal layers of these visceral tissues. From there, the infection can spread through these peripheral nerves up into the brainstem. And so this leads to persistent viral infections, as well as reactivation of herpes viruses that lie in these same nerves and brainstem. And I'd like to emphasize that one of the potential reservoirs for HSV1, again, is the perineurium. And so in the case of COVID and long COVID, we really need to consider perineurial cells as a place where HSV1 can reactivate, and where these human endogenous retroviruses, the herpes viruses, can also reactivate the perineurium. And so here, bringing these elements together, SARS-CoV-2 entry sites are present in all structural elements of the human glossopharyngeal and vagal nerves, clinical implications. And this diagram helps illustrate that you have an organ, in this case the tongue, or the pharynx, or the epiglottis, and these portions of the oropharyngeal cavity and its organs are innervated by different cranial nerves. The 7th, the 9th, the 10th, And each of these nerves has a bundle of cell bodies, and these are known as ganglia. The geniculate ganglion, the petrosal ganglion, the nodose ganglion, and that's part of the vagus nerve. These neurons project into the brainstem, and they influence the dynamics of second-order neurons present in the brainstem. That's how signals are transmitted, relayed to the brainstem, and from there to higher centers. This group of second-order neurons are involved in autonomic functions such as breathing rhythms. For instance, the Butzinger complex and the pre-Butzinger complex. So viral infections, if they're transmitted into the brainstem, this can have severe pathologic damage to the brainstem, or just neuroinflammation of the brainstem can alter the function, the autonomic functions that are controlled by these regions. And this is one of the ways that dysautonomias can be established. Now, this paper published in 2022 was solely focused on SARS-CoV-2, not the reactivation of herpes viruses or the reactivation of HERV viruses, which are both possible as a consequence of a COVID-2 infection moving through cranial nerves into the brainstem. So we're going to take up that particular issue, this pyramid of pathology emanating from a primary COVID-2 infection linked to a secondary HSV1 reactivation to a tertiary reactivation of HERV viruses and the expression of these pathologic proteins involved in neurodegeneration. Now, in part 2 of this discussion, we're going to take up this case series, it's a set of case studies. Clinical activity of Behçet's disease associated with SARS-CoV-2 and herpes simplex virus. Insights from 3 case reports. This is published in 2025. Finally, in closing, I would like to encourage you to visit other YouTubes on this channel. Here is a series that discusses ferroptosis driven by the silencing of the gene which encodes GPX4, glutathione peroxidase 4. And this silencing is mediated by hypermethylation of the GPX4 promoter. And this paper in particular was discussed as a central mechanism by which injuries that occur during COVID and long COVID, which result in ischemic reperfusion injury, could lead to the hypermethylation of the GPX4 promoter, silencing this gene, which is critical for antioxidant, anti-ferroptosis capacity of cells. So if you would like a PDF slide summary of this talk, as well as other talks on this channel, these PDF slide summaries contain active links to YouTubes as well as references on PubMed. Please go to the folder highlighted at the bottom of the slide, COVID therapies. Type in this URL. It'll take you to a Google Docs folder where you can find a set of folders. If you'll navigate to the folder download first, you can find the PDF slide summary to this talk more quickly. So I will pause here and then proceed to part 2 of this discussion. COVID reactivated herpes viruses, human endogenous retroviruses, HERVs, and Behçet's disease, part 2. These are notes for January 29, 2025. In the first part of this multi-part discussion, we discussed these 2 papers, human herpes virus 6, human herpes virus 7, and Epstein-Barr virus reactivation in pityriasis rosea during COVID-19. This was a reactivation of 3 herpes viruses in response to a COVID infection. And this concept of a primary infection, COVID-2 infection, evoking the reactivation of dormant viruses, this can be considered a sequela or a secondary event to the primary infection. In addition, there is a tertiary reactivation of HERVs, ancient or fossil viruses, and that's abbreviated HERV, HERVs. The second paper we discussed were gum recessions, gingival recessions caused by herpes simplex virus in a patient with COVID-19 infection. So this 3-step process, COVID-2 reactivating a herpes virus, in this case HSV1, and the reactivation of HERVs, leading to the formation of lesions. This shows the complexity of the pathogenesis and helps steer the discussion towards various therapeutic interventions. To help focus this discussion in part 2 of this series, I would like to introduce the condition known as Behçet's disease, also known as Behçet's syndrome. It is an autoimmune disease which expresses itself in recurring severe ulcerations of the oropharyngeal cavity as well as the genital region. In the oral cavity, the oral ulcers of Behçet's often contain reactivated herpes simplex virus 1. And so implication, and this is a hypothesis, that associated with these HSV1 reactivations, there'll be a tertiary reactivation of HERVs that are characteristically activated by HSV1. So we'd like to introduce some of this literature. There are over 14,000 papers on PubMed dealing with Behçet's disease, so it is a very useful set of information to compare with other neurodegenerative disorders, autoimmune disorders, and there are a few papers which have been published on what happens when Behçet's patients contract COVID. We will be discussing those as well. So the first paper I would like to mention, Behçet's disease patients with multiple sclerosis-like features, discrimination value of Barkhoff criteria. Neuro-Behçet's disease misdiagnosed and treated as multiple sclerosis, a deceiving masquerader. Now, as I mentioned in the first part of this series, if you look for HSV1 Alzheimer's, you'll find 325 results. If you look for neuro-Behçet's, you'll find 573 results. So Behçet's, even though it forms visible pathologies, namely ulcerations of the oropharyngeal cavity and the genital region, the pathologies can also be expressed in visceral organs and in the nervous system. So let me just read an AI overview of Behçet's disease. Behçet's disease has a strong genetic link, primarily with the HLA-B51 allele, a gene variant involved in immune response, significantly increasing risk but not causing the disease alone. While HLA-B51 is found in many Behçet's disease patients, especially along the historic Silk Road in the Middle East, Central Asia, it is not diagnostic, as most carriers do not develop Behçet's disease, and other genetic environmental factors are involved. The HLA-B51 gene influences the disease's presentation and severity, often correlating with more severe outcomes, especially in younger males, and interacts with other genes like ERAP1. To better understand how CoV-2 reactivates herpes viruses, particularly HSV1, Behçet's patients may serve as a very important human cohort to understand these reactions. As we mentioned in the first part of this talk, the reactivation of HSV1 in the central nervous system has now been linked as one potential driver of Alzheimer's disease. So the downstream events of HSV1 reactivation involve the reactivation of certain HERVs. And so this sequence of a primary insult, in this case a CoV-2 infection, reactivating HSV1, a secondary response, and the reactivation of HERVs, a tertiary response, leading to pathological lesions, Behçet's ulcers with the presence of HSV1 and potentially the reactivation of HERVs in these lesions should serve as a useful system to understand the impacts, treatments that are designed to suppress long COVID symptoms, the reactivation of herpes viruses. Now, I'd like to tie these concepts together. First, what is the HLA gene complex? It stands for human leukocyte antigen. It's located on chromosome 6. It is essential for immune system function by producing proteins that distinguish self from non-self, presenting antigens to T cells and regulating inflammatory responses. These highly polymorphic genes are critical for defending against pathogens, influencing transplant compatibility and contributing to autoimmune disease susceptibility. And again, Behçet's, with its HLA-B51 allele, predisposes people to this condition, which is viewed as a combination disorder, part autoimmune, part infectious agent. And HSV1 is one of the key infectious agents that's reactivated in Behçet oral ulcers. What about lesions found in neuroBehçet's? On PubMed, there are over 1,200 papers on neurological aspects of Behçet's disease. Here are two of them. Neurological involvement by Behçet's syndrome, clinical features, diagnosis, treatment, and outcome. NeuroBehçet's disease, a review. When diagnosing Behçet's disease, the following international criteria are used. Signs or symptoms are awarded points. Ocular lesions, 2 points. Genital aphthosis, 2 points. Aphthosis is a large, non-healing ulceration, like a canker sore. Genital aphthosis are canker sores in the genital region. Oral aphthosis, oral ulcers, 2 points. Skin lesions, 1 point. Neurological manifestations, 1 point. Vascular manifestations, 1 point. Positive pathergy test. Pathergy is when a bump to the skin results in a inflammation or ulceration. These are the clinical features of Behçet's. However, sophisticated neuroimaging methods like contrasting MRI can detect neurovascular lesions in the brains of certain Behçet's patients. These are then diagnosed as having neuroBehçet's. On the upper left, you see a figure from a paper on neuroBehçet's showing a neuropathological features of meningoencephalitis of Behçet's. So this is H&E is hematoxylin eosin staining, 60x magnification, showing an infiltration of lymphocytes around a vein. Now, this type of infiltration and aggregation of white blood cells around a blood vessel is known as perivascular cuffing, and the aggregation of white blood cells is called a perivascular cuff. And these are very common in advanced MS patients, very pronounced in large perivascular cuffs. And so this is a sign of hyperimmune state and cells crossing the blood-brain barrier and accumulating between the blood vessel and the basal lamina to which the end feet of astrocytes are attached. This is in the central nervous system. So in the upper right of this slide, I have illustrated a plausible three-step set of events that could produce a perivascular cuffing. One possible scenario by which it could occur in a long COVID patient or a Behçet's patient. CoV-2 would be the primary infection. It reactivates a herpes virus, HSV1. HSV1, in turn, upregulates HERV-K protein, the expression of a HERV virus, an ancient or fossil retrovirus. And this HERV-K protein is immunogenic and pathologic, toxic. The presence of HERV-K protein could lead to a perivascular cuffing. Now, I should mention that perivascular cuffs are not just lymphocytes. They are also filled with leukocytes, including neutrophils. And so, in the discussion of long COVID perivascular spaces, you need to distinguish what types of cells are in these perivascular cuffs. For neurologists and neuropsychiatrists, the presence of persistent perivascular cuffs is a sign of neuroinflammation. Earlier on this YouTube channel, I reviewed the literature on perivascular spaces in the context of long COVID. Persistent and large perivascular spaces, which are detected through MRI, these are part of the lymphatic system, and these 5 YouTubes discuss how infections of the brainstem by viruses, including CoV-2, can result in the enlargement of perivascular spaces. So the perivascular spaces are important from a diagnostic point of view, but also from a mechanistic point of view of understanding neurological disorders such as dysautonomias, which occur as sequelae to the primary viral infections. There are 3 YouTubes on this channel which deal with enlarged perivascular spaces in the context of long COVID. Those are highlighted in green at the bottom of the slide. Now, utilizing AI overview to look at the information about specific HERV virus proteins in the context of neurological disorders. First, looking at multiple sclerosis. Human endogenous retrovirus K, HERV-K, specifically HML2 envelope, and that is abbreviated ENV, and GAG proteins are increasingly recognized as potential pathogenic factors and biomarkers in multiple sclerosis. While HERV-W is more commonly associated with MS, HERV-K expression is also found in MS, with recent sensitive mass spectrometry techniques, often using membrane-enriched samples, detecting these proteins in cell lines, and potential links to immune activations. Key finding on HERV-K in MS. Presence in MS. HERV-K GAG and envelope proteins have been detected in MS patient samples. Pathogenic potential. Similar to HERV-W's syncytin-1, HERV-K is believed to trigger inflammatory responses. Now, in the upper right of the slide, I have placed a H and E stain, hemotoxylin and eosin histological stain of a blood vessel. This was taken as a biopsy from a patient with neurobechettes. And once again, there is an arrangement of white blood cells, in this case leukocytes, surrounding the blood vessel. These are the blue dots. And this aggregation of white blood cells, these are white blood cells that left the bloodstream, crossed the blood vessel, and entered the perivascular space, forming a, quote, perivascular cuff. This is similar to what is seen in multiple sclerosis. This is a conjecture, hypothesis, that the HERV proteins that are secreted in response to the reactivation of dormant viruses, such as herpes simplex virus 1, or other aspects that turn on the promoter of the HERV virus gene, encoding these proteins, whatever is driving the promoters of these retrovirus genes, these ancient fossil viruses, the presence of these proteins activating the immune system, the histological clinical finding could be an enlarged perivascular cuff. That's not to say that all perivascular cuffs would necessarily contain HERV proteins, but in the case of certain neurological disorders such as MS, ALS, Alzheimer's, perhaps long COVID as well, the presence of these HERV-K proteins may be a key element which is promoting the extravasation and accumulation of the white blood cells, both lymphocytes and leukocytes. Utilizing AI overview to look at HERV proteins in the context of amyotrophic lateral sclerosis, ALS. Human endogenous retrovirus K, HERV-K envelope protein, and RNA are detectable in the cerebral spinal fluid of a subset of patients with neurodegenerative diseases, particularly ALS patients, as well as Alzheimer's disease patients. The expression of HERV-K in the central nervous system is linked to neuronal damage, with its envelope protein acting as a neurotoxin that may cause motor neuron degeneration. Key findings on HERV-K in cerebral spinal fluid. ALS pathogenesis. Studies have identified HERV-K HML2 envelope protein and RNA in the cerebral spinal fluid and brain tissues of ALS patients. Neurotoxicity. NERV-K envelope protein is directly neurotoxic, causing the retraction and beating of neurites, projections from nerve cells, in human neuronal cultures. Mechanism of action. The protein induces mitochondrial dysfunction, increases reactive oxygen species, and causes cell death. It is believed to be regulated by TDP-41, a protein often associated with ALS. A recent paper from 2022 is shown at the lower right of the slide. Human endogenous retrovirus K, HERV-K envelope protein in spinal fluid of amyotrophic lateral sclerosis is toxic. So we utilize this slide in the first part of this YouTube series. I'd like to again emphasize that a set of viruses that can provide the primary infections can reactivate these dormant fossil viruses known as human endogenous retroviruses. And when these ancient viruses are reactivated from the human genome, they express proteins, which are frequently toxic and immunogenic. So the key is, what are the transactivators of HERVs in the context of diseases such as long COVID? That will require an analysis of the promoter regions of these ancient viruses. And the metabolic pathways that cells use to reduce reactive oxygen species may be part of the key to understand when HERV viruses reactivate. For instance, under oxidative stress and the excess production of reactive oxygen species, ROS. These pathways, which are promoting cytokine production, may be the triggers for the reactivation of these HERV viruses. And again, would like to emphasize that during viral infections, autoimmune diseases, and cancers, frequently there is a methylation of genes, such as the vitamin D receptors promoter. And there's also the methylation of the GPX4 promoter. And GPX4 is a critical enzyme to deactivate reactive oxygen species as well as peroxidated lipids. And so in cells that have compromised expression of GPX4, this may predispose them to the reactivation of these HERV viruses. And as a consequence of that, a sequela to the reactivation of these HERV viruses, the production of these toxic HERV proteins could initiate changes in cell behavior, which will ultimately manifest in the production of histological lesions such as enlarged perivascular spaces. Now connecting these concepts to COVID infections. This paper from 2023, SARS-CoV-2 awakens ancient retroviral genes and the expression of pro-inflammatory HERV-W envelope protein in COVID-19 patients. Recent studies have identified a significant link between SARS-CoV-2 infection and the activation of human endogenous retroviruses, HERVs, particularly HERV-W envelope protein, ENV, which is suspected to contribute to the severity of COVID and the persistence of long COVID symptoms. A sidebar comment. I would like to again emphasize the potential importance of looking at the reactivation of herpes viruses and HERVs viruses in Behçet's oral ulcers in the context of COVID-2. We will be discussing recent papers where Behçet's patients have been infected with COVID-2 and what happens to their physiology as a consequence. However, in terms of the virology and molecular cellular dynamics of lesion formation, the Behçet oral ulcer may be a very accessible site to study how COVID-2 reactivates HSV1 and then subsequently HSV1 reactivating HERV viruses and their toxic proteins, how all of this may contribute to the persistent ulceration that is found in Behçet's oral ulcers. Now, this is a graphical abstract from the paper we have just been mentioning. SARS-CoV-2 awakens ancient retroviral genes and the expression of pro-inflammatory HERV-W envelope protein in COVID-19 patients. I have condensed what is shown in the graphical abstract of the paper into a linear information vector. COVID-2 reactivating HERV-W directly. There's no herpes virus reactivation involved in this step. Once the HERV-W virus is reactivated, there is expression of the HERV-W envelope protein. And the HERV envelope protein leads to tissue pathogenesis. Now turning to Alzheimer's. Repetitive injury induces phenotypes associated with Alzheimer's disease by reactivating herpes simplex virus 1 in a human brain tissue model. These are organoids. Reading from the abstract. These data suggest that HSV1 in the brain is pivotal in increasing the risk of Alzheimer's disease. as other recent studies using cerebral organoids have suggested. We propose that after brain injury, whether by infection or mechanical damage, the resulting inflammation induces HSV1 reactivation in the brain, leading to the development of Alzheimer's disease/dementia, and that HSV1 is a major cause of the disease, especially in APOE carriers, APOE carriers. This does not preclude the existence of other mechanisms in the etiology, the causes of Alzheimer's disease, particularly those involving other infectious agents. Future studies will investigate possible ways of mitigating or stopping the damage caused by head injury, such as anti-inflammatory and antiviral treatment after injury, thereby preventing HSV1 reactivation in the brain and reducing subsequent development of Alzheimer's disease. A sidebar comment. On this channel, we have been emphasizing that active vitamin D receptor agonists, not vitamin D3, hydroxylated versions of vitamin D, particularly calcifediol 25 vitamin D3 and paracalcitol 125 vitamin D2, used in tandem, could be potentially used to suppress HSV1 reactivation or suppress reactivated HSV1 reactivation in various IAC conditions, or perhaps in the case of traumatic brain injury, TBI, which this paper now suggests is a key driver for the reactivation of HSV1, which may lead to Alzheimer's disease. I would like to draw your attention once again to this paper utilizing active vitamin D hormones, calcifediol, the liver metabolite of vitamin D, which does bind vitamin D receptors and activate them. It's at a concentration of about 1,000 times more concentrated than the kidney metabolite of vitamin D, calcitriol. Calcifediol, its normal reference range is 30 to 100 nanograms per mil. In contrast, calcitriol is about 1,000 times less. Its reference range is 20 to 80 picograms per mil. Calcitriol as a supplement has a side effect that is unwanted. It's deleterious, potentially deleterious. Hypercalcemia. It elevates serum calcium two ways, mobilizing calcium release from the bones, as well as increased calcium uptake through the gut. For that reason, nephrologists and endocrinologists have developed an important synthetic analog of calcitriol, paracalcitol, which has much less calcemic effects. For that reason, paracalcitol is now the standard of care for chronic kidney disease patients who lack adequate levels of calcitriol, so they supplement with paracalcitol. This was the motivation for combining these two for the treatment of this patient with severe HSV1 infectious keratitis. The keratitis, the infection and inflammation of the cornea was so severe, the cornea perforated and the iris began to protrude out of the perforated cornea. The patient was hospitalized, treated with standard of care for HSV1 infusions of acyclovir. She was also treated with agents to reduce the osmotic pressure of her vitreous humor to allow the iris to move back inside the corneal boundary. A fetal amniotic chorion membrane was grafted to the cornea. Again, this is standard of care for these types of infections. And she was then started on calcifediol and paracalcitol after she was released from the hospital. While she was being treated, first with a combination of calcifediol and paracalcitol, there was no recurrence of her HSV-1 infectious keratitis, which often occurs in these cases. In addition, the cornea began to heal. At a couple of months after treatment, the pericalcitol was removed, but she was maintained on daily calcifediol. Her cornea began to heal and thicken, but she was left with a corneal scar. You can see that in the right panel. About a year after she was released from the hospital and healed cornea, she had a corneal transplant, and she recovered about 80% normal vision in this eye, a remarkable outcome. So combining this important paper on the use of active vitamin D hormones to treat HSV-1 infection in the cornea with this paper on repetitive injury inducing Alzheimer's by reactivating HSV-1, reading again from the conclusion of this paper, future studies will investigate possible ways of mitigating or stopping the damage caused by head injury, such as anti-inflammatory and antiviral treatment after injury, thereby preventing HSV-1 reactivation in the brain and reducing subsequent development of Alzheimer's disease. So the methods that were used to treat this case of severe infectious HSV-1 keratitis could potentially be translated to patients who have recurring TBIs, or in the case of long COVID, one could conceive that the chronic infections and viral reactivations in the central nervous system of people with long COVID, that again, the use of active vitamin D hormones used in combination could be potentially used as adjunctive therapies with agents like acyclovir to suppress HSV-1 and the associated tissue damage. On this channel, there are a number of YouTubes describing the potent activity of pericalcitol, its ability to suppress ferroptosis. Ferroptosis is a specific form of programmed cell death associated often with viral infections as well as ischemic reperfusion injuries. And both of these pertain to the ontogeny of Alzheimer's disease. So in closing, I would like to summarize the concepts that have been presented in parts 1 and 2 of this multi-component YouTube series. So here is a graphical summary. We're looking at a set of sequential viral transactivations. We can abbreviate that by SVT. First, step 1, a COVID-2 infection reactivates herpes viruses, such as HSV-1 and or Epstein-Barr virus. These are just two of the herpes viruses that can be reactivated by COVID. Step 3, HERV proteins can be reactivated either by COVID-2, HSV-1, or EBV. These HERV proteins are toxic and immunogenic. So besides the HERV proteins, there are multiple proteins and RNAs being generated by these upstream viral infections. And so what is a result of this is a combined viral pathogenesis, all arriving from a process which we can refer to as sequential viral transactivations. We can utilize the concept of sequential viral transactivations to introduce a key paper which we will examine in more detail in part 3 of this YouTube series. HSV-1 and endogenous retroviruses as risk factors in demyelination. And this paper will be used to analyze demyelination events that are occurring in a subset of long COVID patients. So reading from the abstract of this current paper, herpes simplex virus 1 is a neurotropic alpha herpes virus that can infect the peripheral and central nervous systems, and it has been implicated in demyelinating and neurodegenerative processes. Transposable elements are DNA sequences that can move from one genomic location to another. Transposable elements have been linked to several diseases affecting the central nervous system, including multiple sclerosis, a demyelinating disease of unknown etiology influenced by genetic and environmental factors. Endogenous viral transactivators may activate certain retrotransposons or class I transposable elements. In this context, several herpes viruses have been linked to multiple sclerosis, and one of them, HSV1, herpes simplex virus 1, might act as a risk factor by mediating processes such as molecular mimicry, remyelination, and activity of endogenous retroviruses. Several herpes viruses have been involved in the regulation of HERVs, and HSV1 in particular can modulate HERVs in cells involved in multiple sclerosis pathogenesis. This review exposes current knowledge about the relationship between HSV1 and human endogenous retroviruses, focusing on their contribution as a risk factor for MS. Now, looking at the information landscape about virally induced demyelination on PubMed, if you search for demyelination and long COVID, there are about 240 results. In 2025 alone, there are 25 papers on this. HERV and demyelination give you 236 results. 2 key papers. Role of demyelination in the persistence of neurological and mental impairments after COVID-19. Multiple sclerosis and SARS-CoV-2 has the interplay started. Again, looking at this material through the lens of a process, sequential viral transactivation, starting with the COVID-2 infection, reactivation of herpes viruses, reactivation, transactivation of HERV viruses, and the production of toxic, neurotoxic HERV proteins, producing demyelination. And here is one of the figures from the paper we will go into more detail in part 3 of this discussion. Role of HSV1 in HERV's activation and immune response. The figure summarizes relevant effects of herpes simplex virus 1 on HERV's transcription and the synergistic effects of both viruses on immune response. Again, the concept of combined viral pathogenesis arising from sequential viral transactivation. If you would like a PDF slide summary of this talk, please go to the Google Docs folder highlighted in green at the bottom of the slide. Here you will find a PDF slide summary of this particular YouTube, which contains active links to other YouTubes on this channel, as well as certain references on PubMed. If you navigate to the subfolder Download First, you'll be able to locate this PDF more quickly. At this point, I will pause, and then we will go on to part 3 of this discussion.